Hello everyone, thank you for checking out my poster, and thank you to UCR for allowing me to present during this crisis. My project is on developing methods that are looking at the regional variations in vasculature structure of the gubernaculum and how it might provide insight on unknown cryptorchidism mechanisms. My name is Human Monajemi, and I'm working with Dr. Joshua Morgan from the Department of Bioengineering. Cryptorchidism, or undescended testes, is the most common congenital structural defect and can lead to a significant increased risk for testicular cancer later in life. Although previous research has shown androgen receptor expression to be altered in the gubernaculum for rats that are susceptible to cryptorchidism, the mechanism that links the regional differences is unknown. The gubernaculum is a specialized tissue that aids in the process of testicular descent through the changes in its structure throughout infant development. The organ can be divided into three main parts: the tip, the mid-portion, and the base. Now, we know from previous research that androgen receptor expression regulates angiogenesis. Therefore, we hypothesize that there are changes to the vasculature structure of the gubernaculum for the rats that are susceptible to cryptorchidism. Previously, the segmentation of the tissue was done manually through the use of a mask observer, and the different regions were identified by hand. This can be a very tedious procedure for large stacks of 3D images and may introduce bias in regionalization. My aim is to develop custom MATLAB scripts that utilize computer vision techniques to segment the tissue and identify the regions based on the structure geometry. To develop this custom MATLAB script, there are four main steps highlighted here. First, we would like to identify the tissue's region of interest and isolate unnecessary areas using a deformable model with active contours. Next, we would like to segment the tissue by geometrical regions through an analyzation of the tissue's curvature. Third, we would like to implement vasculature segmentation using a Frangi-Hessian hybrid filter. And finally, we are able to compare the regional vasculature structure of the control groups to the mutated groups. To elaborate on the methods, we can start by looking at figure 1a. Here we see a slice within the 3D stack that shows the gubernaculum with minimal noise. But looking at figure 1c from the same stack, we can see the formation of the gubernacular cord highlighted by the green arrow. Since we are not looking into the properties of the gubernacular cord, we would like to omit that structure from our quantification. This needs to be done manually for every stack of images, but using active contours, we are now able to segment the structure in figure 1a as seen in 1b and use its boundaries to create a deformable model that adapts to the rest of the stack. This model expands and shrinks with the tissue structure but leaves out extreme changes such as the cord. You can see the model highlighted in red outlined in both figures 1a and 1c. To divide up the structure, we need to determine the extreme points that help us identify the different regions. In figure 2a, the top point, considered the base point, is calculated based on the max distance away from the boundary, or basically the furthest point from any edge within the tissue. The bottom point, or the tip point, is a boundary point that is the furthest away from the base point. Using these 2 points, the shortest line that can connect them is calculated based on the curvature of the tissue. Looking at figure 3, 15% of the shortest line from the tip point to the base point is attributed to the tip region. Everything about the base point is identified as the base region, and the area in between is considered the mid portion, which is further divided into the inner and outer curvature. Finally, we can use the Frangi-Hessian filter to segment out the vessel structure within the gubernaculum. Then the vesselness can be quantified based on the region of the gubernaculum that it is located within. In conclusion, we have developed a novel algorithm for automatically segmenting regions of the gubernaculum. Future work will apply this algorithm to quantify regional differences within the gubernacula and incorporate more advanced statistical methods. The authors would like to thank Dr. Julia Barthold, Dr. Alan Robbins, and Dr. Gray Dion for providing the dataset. We would also like to thank the UCR Undergraduate Education for funding this work with a mini grant.